Welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
And I'm Sylvia Earle, an oceanographer, National Geographic explorer, and founder of Mission Blue.
And an ocean elder.
Oh, and an ocean elder, of course.
Of course.
Thank you, ocean elders, for hosting this extravaganza.
It is an extravaganza most times too. And this is our show, Dive In, where we host informal and open conversations with the ocean community on topics of wonder and interest. And as always, the topic of wonder and interest is, will I be able to share the screen? Host is disabled sharing. Uh-oh. All right, well, wait 1 minute. And in the meantime, we're going to let everybody know that later on in the session, we're going to take questions and answers towards the end, and we'll be posting some links in the chat box for people to access. And Gigi, if you can allow us to share the screen, that would be awesome, so that we can remind everyone that the world is blue.
The world definitely is, especially when viewed looking at the blue face of the planet, the Pacific, where the land seems really trivial. But of course, there's land underneath the blue, but it's the ocean that really sweeps over the surface that makes our existence and the rest of life on Earth possible.
And I particularly like this view of the Earth because it shows the blue back of the planet, the blue shoulders that hold it all together. Okay, so today we're being joined by our longtime friend, Susan Brawley. Susan is a marine ecologist at the University of Maine, where she's been a professor since 1994. And she earned her PhD at Cal Berkeley. Go Bears.
That's what I thought you said.
She's renowned for her work with algae. There she is. Thanks, Susan, for popping in without me having to say anything or do anything. She's a fellow of the American Association for the Advancement of Sciences and received the Award of Excellence from the Phycological Society of America in 2020.
It's phycological, not psychological.
Right? And how often does that happen? We don't need the, the fainting couch or anything like that. We need we need the kelp forest.
Yeah, the so-called seaweeds. Kind of an all-embracive term.
Yes.
Mostly algae.
Mostly algae. Well, welcome, Susan. Thank you so much for joining us today.
It's a pleasure. Thank you.
Yeah.
Fun to talk with you.
Yeah, and we have a lot of slides that you have lined up for us, which is awesome. But to start off with, tell us, I know you two have gotten into a lot of algae mischief over the years. So how did that all start out?
So a guy named Harold Hum really started it. He was a very fine phycologist.
There he is with some seaweed.
Yeah, that's right.
Yeah.
And so he was Sylvia's PhD advisor. He was always very good to do things with children. And you actually met him long before you actually did your PhD with him. Is that correct or not?
He taught a summer class at Florida State University, and I was allowed to take it as an undergraduate. All the others were graduate students, but it was, I think, because I already had fallen for life in the ocean and he encouraged that. And later, I mean, just one thing after another, he guided me through both a master's and a PhD. But he got to you when you were even younger than when I first met him. Weren't
yes,
you like
when I was 12 years old.
Yeah.
And so he led a trip for the Charlotte Nature Museum to the Duke Marine Labs, where he had been the associate director for a long time. And it was such a wonderful field trip. I was already interested in the algae in the sea, and we streaked out bioluminescent bacteria. We collected algae and identified them and pressed them as herbarium specimens. I still have them. And I helped him lose a phytoplankton net. I can still see him rowing. And all of a sudden, the phytoplankton net came off its tether and we just watched it going off.
Here.
Oh,
No.
Another
But
piece of
anyway,
scientific equipment gone.
yeah. So he invited me to work in his lab at Queens College, which is where he was then, on Saturdays. And so my parents would take me in, and we would do a lot of culturing of bacteria. And I would take tubes home and work on them in my lab during the week and come back. And we would do things, to advance the next step of helping me understand the important roles of marine bacteria. And he showed me how to extract agar from seaweeds and things like that. So it was just
Make your own ice cream.
and
And
then
toothpaste.
he went off to the University of South Florida, and I visited him several times there and went off on trips to the Keys. So then when I was coming to Boston to college, he wrote you to tell you that I was coming. And that's how we met. It was so lovely.
It's the power of a sympathetic teacher.
Yes.
I mean, if you mark points in your history, why did you start down this path, or who in all of those you met have been most influential? Well, in your life and mine, Harold Hum really looms large, but we aren't the only ones. He used to take there. I mean, he was a university professor, for heaven's sakes, but he, as you said, he had a passion for kids. And in his summers, when many university professors hunker down over hot microscopes and turn out their learned papers, he would often work with high school teachers and high school students. I remember seeing a school bus filled with these students, and they had a big banner on the side. His name is Harold Hum. They call themselves the Humdingers.
Well, and you found an algae that you named after him. You called
I
it
did. It's called-
umbrella,
I
right?
did. Here it is. It was, we had a little discussion at Duke about what to call it, but because this little beast, it's a red alga like your Porphyra there. This picture is in black and white, but we thought it kind of resembled an inside out umbrella.
It's
So of
true.
course, we called the thing Humbrella. It's Umbrella Hydra. Anyway, yep.
Yeah, I
It's
knew you
history.
two were just going to launch off.
Yeah. Take it away, Susan.
But tell us, like, how many kinds of algae are there? It seems like there's just like a babillion kinds, and are they closely related, not related at all? What's up
Well,
with that?
most of them are not closely related. And that's the really amazing thing we found out over the last 30 years because of the advent of molecular sequencing. So just as we can find out our ancestry from very recent connections to very deep connections, we can do the same thing with the algae. And so, if you go to any of the textbooks that we used when we were in high school and college, they would say that all the algae had a common ancestor. And so this is a diagram that shows what are called eukaryotic supergroups now. So of course, there are bacteria which don't have their DNA in nuclei. It's just loose within the bacterial cell. And then nucleated cells can either be unicellular, like diatoms, for example, or paramecium, or, think of any one you want to, or they can have evolved to be multicellular, like us. And so one of the things that's just amazing about what we've learned in recent years is that, can you see my pointer?
Yeah. No,
So
well,
this is a supergroup of land plants and algal relatives, namely a unicellular group called glaucophytes, and then the red algae. I can talk about this in a second, maybe.
yeah.
And they're the green algae with their little branching group, the land plants, right? So the land plants have all evolved from part of the green algae. And so this is an incredibly important group because this is the point at which nucleated cells had captured a photosynthetic bacterium, which we
Incredible. It is.
know as a cyanobacterium. And so this is extremely important to our planetary system now. And this is when the Earth really started to be oxygenated, both by the evolution of photosynthesis in cyanobacteria billions of years ago, and then possibly as long as 2 billion years ago, the evolution of these nucleated cellular algae that had chloroplasts, because the endosymbiotic cyanobacterium was really co-opted by the cells that had captured it. And they, that was great.
When Lynn Margulis suggested that this was really going on, I mean, she's a woman after all, and her ideas were scorned. It's only later that people adopted it and understand that, Darwin was all about competition and survival of the fittest. Lynn Margulis and others have come to see that cooperation, collaboration, co-opting bacteria to become integral to the cells of other creatures. I mean, it's a very refreshing way to look at the world, that more his progress has occurred because of collaboration, working together, than fighting one another. I mean, there is some of that, but progress comes.
Yeah, actually, and one of the interesting things about that is that although Margulis didn't know about him, and as you've described, she really had to fight for her ideas to be recognized and accepted. Marinchowski, who was a Russian botanist who got interested in lichens and therefore started being interested in this cooperation between different organisms, he really is the first person to state the ideas of endosymbiosis concretely in one of his early papers. And what you have just said reminds me about the way he ends it. And he said, imagine how important this concept is and how much it has changed evolution of different groups of organisms. So a lion under a palm tree, and if this lion had only captured these little green photosynthetic organelles, it would just lie down peacefully under the palm tree and only need to sip mineral salts, right?
I've always wanted to photosynthesize, it would be so cool to be green.
I think that's actually one of the most profound things that's ever been written in a scientific paper, because the different evolutionary paths in which plants and algae became photosynthetic and could make their own food kept them from having to go out and catch other things. And I mean, you can just imagine that the whole concept of war.
Yeah,
that's the difference, right? Of organisms that go to war and organisms that just sit in the sun. I'm not talking about Venus flytraps, of course.
That's true. That's true. But it is incredible how many of the algae are used for food, though. We do use them. We go out and gather them. I found this package of
wonderful.
Yeah,
yes.
Right.
Yeah.
And manning my chopsticks here, I found these rice crackers, right, that are wrapped up with nori. Here you go. Oh dear.
Like one? I wish I could offer to you. There we go.
You're making me hungry.
Yeah, but I mean, it just comes like as this dried vegetable in the package, but the sea vegetables are just like this should really be what we think about when we think of seafood, right?
Yeah.
And not a fish, but it's just such an interesting thing. Wait, I think I've got a couple. Here's some of the sheets of it, right? It's
Yeah,
kind
the
of
sheets
dried.
of Nori. That's right. So that's a Porphyra relative, now called Neopirophia.
And then we enjoy it in our veggie sushi.
Veggie sushi rocks.
I really am getting hungry, and I'm sure our friends are listening are too.
That's right. And you've really been advocating for a lot of this algae as a food, and all of the micronutrients that are involved in it.
Yeah.
So it just seems a really good way to get all those micronutrients. And I think that one of the things that is really impressive about it is the DHA, or the algae oils.
Many of the ones that are available in pill form, the algae derived ones come from dinoflagellates. That's how the fish acquire their oils, by eating them. They don't manufacture the oils or synthesize them. They come in their diet. So we don't have to munch on fish to get so-called fish oil. You can do what the fish do. Start at the bottom of the food chain.
And then this is you sent this slide of like all these different sea vegetables. What have we got laid out here? You talk about some of the different kinds that are edible.
Well, I think that about half of these dishes do have sea vegetables in them, but I'm pretty sure that is a meat dish sitting at the very center. This was a banquet when I left Qingdao, China, after working there for 6 months in 1984 in Professor Fei's group. And right in the center on the left is Professor Li and Chen Mei Chen, who was one of my best friends there. And so, a banquet in China has 20 dishes, and everybody made them and brought them, and we had really fun. But some of these would have had nori in it. Some would have had gracilaria, which is a red seaweed. Some would have had kelps.
Yeah.
And so we really did feast. It was delicious. And of course, in Asia, typically people put together dishes in which there's a little bit of a lot of different things. So it makes it very healthy to eat.
Very complete in a small package.
No vitamin pills needed.
No, not at all. So what percentages of the macroalgae that are used for food, how much is like actually just gathered from the wild versus being raised in aquaculture settings?
It really depends in part upon whether you're talking about traditional harvesting or aquaculture. And then there's some companies, for example, the Dulce package you showed and the Laver package. It's a Maine company called Manco Sea Vegetables, and it's the oldest in Maine. And they have been very supportive of the development of seaweed aquaculture, but they sustainably wild harvest for a lot of their crops still too. I don't know exactly the proportion. At this point, they buy from aquaculture as much as half. And I might add, Liz, that their dulse is delicious, eaten as a snack right out of a bag. Feel free.
Good. Here, wait, you've got it back here. This one says wild Atlantic sea vegetable.
Yes.
Okay, here we go.
This is yummy. It is salty though.
Well,
But it's not sodium chloride, it's potassium chloride. And so that's the other thing that's great about eating sea vegetables. You can get a salty taste, but it's not going to give you sodium. And so it makes it very healthy.
Yeah, no, that's a really nice flavor.
So shupu was one of Faye's students and also C.K. Sung's. And this shows the Porphyra and Nori life history. And so the commercial part is over here with these blades. And so this is what we either grow in aquaculture or wild harvest. And in a normal Porphyra life history, there's fertilization of an egg, and then it cleaves up into spores, and they are released into the seawater. And then remarkably, they bore into shells. And this
Wow.
is the form called a Conchocoelus. And it's called Conchocoelus because it had been described as its own genus, separate from Porphyra. And people didn't think they were closely related at all. They were even in different orders.
They look so different. Little filament and then a big sheet.
Big leaf,
Yeah.
yeah.
That's right. So a couple slides on, I have, yeah. So
Oh,
this
there.
shows the Conchocoelus growing in the shells. And
Mhm.
so commercial cultivation is worth a lot of money worldwide. And in order to seed the nets, they change the temperature and light conditions, and conchospores are released from this phase of life history. And then they have a big thing that's almost like a Ferris wheel with nets draped over it, goes into a bath, and the spores stick right away. And then you put the nets out into the sea, and they are growing up little blades.
How cool is that?
Yeah.
Wow.
So here is Kathleen Drew
Famous
Baker.
Psychologist.
Another
Yeah.
phycologist.
Right. And along with Sylvia, probably, the most famous psychologist. So Kathleen Drew Baker was just fascinated by the algae, and she was studying life histories of a lot of different species. And so she realized that the Conchocoelus was actually the other phase in the life history, the diploid phase, compared to the blade gametophyte phase. And this was published in Nature, which is the top journal in science still. And it was published there in 1949. Well, we didn't have email or anything, and she knew how important this discovery would be to her Japanese colleagues. So she wrote them and told them about it. And they immediately repeated her work on their species. And within just a few years, commercial aquaculture was starting in Japan. So she is directly responsible through her basic science for starting this immensely important and valuable and tasty industry. And so in Japan, they have a festival to her every April on her birthday. And this was on the centenary of her birth. So they invited her kids over from England, and they brought her academic robes and draped them on this memorial sculpture that the Japanese fishermen had put there for her. It's a little park in Uto City that commemorates Kathleen Drew Baker. Isn't it great?
It is great. Science respected. What a concept.
And the fact that, just say it again, that women scientists have not been have had a bit of a hard time being recognized on a par with their male counterparts. You're one of the wondrous exceptions, Susan, and so was the Porphyra queen.
And fortunately, it's getting much better. It's already
That's
much better, right? Much
Yeah.
better.
Much better. And what's going on in this image here? We get like the harvesting.
So these photos were given to me by Kiyosuke Miwa, who took me out on these grounds when I was in Kobe, Japan, for an international seaweed symposium once. And so they devised this wonderful system in which just two fishermen can pull on a yoke and immediately pop up all these nets. And they are left out of water for several hours. And what that does is to kill the diatoms that otherwise would coat the growing nori. It kills any other kinds of invertebrate larvae that would otherwise settle on the nets. Because the Porphyra, and in this case, the Nori, which is now called Neopirophia, they are extremely tolerant to desiccation stress. So desiccating the nets is an important part of producing a very healthy crop. And at the end of the day, they just release the yolk and poof, all the nets fall down on the surface of the sea.
I was just observing that there is such a thing as the International Seaweed Symposium. Some of the people who are listening might say there's a what?
What?
Right, yeah.
There are, I mean, you just have to get out there and see for yourself. Seaweeds rock. They're
Yes, they
just
do.
great.
I
And
think I remember you had a bumper sticker at some point that said seaweed powered.
yeah, yeah. Salute
Something like
seaweed,
that.
yeah.
And then,
Another famous one is I break for algae.
yes,
If
yes,
you don't have that one, I'll send you one.
yeah.
So isn't this boat cool? So they also designed these special low boats that could dip under the net at one end and run under it. And there's a cutting blade right up here at the top. And so as the net passes over, the blades get cut off and they just fall down into the boat. And enough of the blades are left so that they will regrow, and they can usually get as many as 5 harvests out of one seeded net.
They just simply grow back.
That's right. It's
Grow
incredible.
grow and mow.
Yeah.
Okay, so tell us about this. Is a CK saying he was really instrumental for both of you guys.
I got to meet him in 1973, and he said it was the first time he had been able to speak English in like 25 years. There was, of course, the Cultural Revolution that really caused China to close its doors to most of the world. But he had a PhD from the University of Chicago and Scripps. He was the first diver at Scripps University, Scripps Marine Laboratory at the University of California in San Diego. But then when he went back, the doors shut. Really, he could not communicate after he left the United States in the I think it was 1945.
It was somewhat later, but it was before the, I mean, he actually thought that the communists were going to change things for the better. So he deliberately went back. Scripps had offered him a permanent job, but he wanted to go back to China and help the people form, a modern country. And incidentally, he studied with William Randolph Taylor for his PhD at the University of Michigan.
It was Michigan, right? Not
Michigan,
Chicago.
yeah.
It was University of Michigan, right?
And so I met him at Berkeley because, let's see, when would that have been? It would have been about 1976. He was one of the first 7 Chinese scientists who were sent overseas when things started to open. And they met with President Ford at the White House, but they came to Berkeley first, or at least CK did. And so
he came to the living room here too.
Yeah, he came to the house. I think we actually had some sea vegetables.
Yeah.
As I recall.
No, he was brilliant. Yeah.
Yeah, and I know it meant so much to him to see you. So he is the person who more than anyone else started the immensely large and successful algal aquaculture industry in China. He domesticated Saccharina japonica, which is the sugar kelp that they grow there. And started working on other crops and brought them into aquaculture. And I had met him at several international meetings, and I was interested in trying to use these tiny little crustaceans called amphipods to keep some of the algal crops cleaner that don't have the desiccation tolerance of Porphyra, for example, Gracilaria. And they can get epiphytes in aquaculture. So he was very interested in my ideas. And one of his key colleagues and students was Fei Shugung, who's shown over here with me on the left. And he had just spent a year at Santa Barbara and came back. So I joined his group for 6 months, and I did those kinds of experiments. Really fun.
And it was a different China than the China of today, for sure.
Yeah.
More bicycles and fewer automobiles.
Hardly any automobiles. Yeah.
Yeah.
And what is this algae we've got going on here? This is
So this is Alaria esculenta, and it's growing on Springtide Seaweed's farm in Frenchman's Bay, Maine, which was founded by someone named Sarah Redmond. And they have really done a tremendous job developing an algal aquaculture industry here in Maine. And now there, I think probably as many as 60 companies, some very small and others larger, that are growing seaweeds. So the production off Maine has just continued to climb every year. And this Alaria is particularly nice because it's sort of an unusual kelp. First of all, it's very tasty. Now, all kelps are nice, but this one is really tasty. And it has about 20 to 25% protein dry weight.
wow.
A lot of vitamins and micronutrients and so forth. So I consider it a particularly good kelp to grow. And so I'm glad that Springtide Seaweed and others are doing so.
And it's native where?
It's native in the North Atlantic. And actually,
Great.
that's sort of an interesting story because a young Canadian, Trevor Bringlow, has just published a really interesting study of all the alarias in the circumpolar Arctic and in the North Pacific and the North Atlantic. So at one point, everything was called Alaria esculenta, but now we know that the alarias that you would find on the Washington State coast and British Columbia and so forth are Alaria marginata. And in the Atlantic, we have Alaria esculenta. And Trevor thinks that there is a cold water circumpolar new species of Alaria.
Nice.
His work. So he's got to do some more work before he describes it, but
Are they all tasty?
You know,
Are
I
they
don't
all-
know, but I think we should mount an expedition and find out, don't you?
Yeah.
We don't have to pack a lunch. It's going to be right there for us.
That's right.
That's right.
That's right.
So this is my friend and colleague Shu Pu, who unfortunately died a few years ago at a too young age, and Fei Shugung, who now looks older, as you can see. But we were off to visit aquaculture farms, and they were helping me figure out how to do some of the things I wanted to do back in Maine.
Excellent.
It was a lot of fun.
Look at this. This is a pretty large aquaculture installation here.
Yes, and these are nori nets. And I should really say that in Chinese, rather than nori, we would say zizai. And I'm sure my Chinese pronunciation is awful, but it's called, by its Chinese name. So at any rate, the nets are strung on bamboo poles rather than being raised by fishermen. But this is a place where the shore goes out for a long, long way with a very gentle slope. So that means if you position the nets on the poles correctly, the tide rising and falling does the work of desiccating the nets to get rid of all those invertebrate larvae and diatoms and so forth, so you can make a good crop.
Yeah, working with nature.
Work with the system. It
Mhm.
seems very simple, right? ingenious.
It's like a piece of art too.
Yeah.
So all of this led me and about 55 other scientists across South America, North America, Europe, and Asia to put in a proposal to the Department of Energy's Joint Genome Institute to sequence the Porphyra genome. And it was one of the first seaweed genomes that was done. And we published it in 2017 in the Proceedings of the National Academy of Sciences. So we had a wonderful meeting that Alison Smith sponsored in Cambridge, England. And so here are some of my buddies on the cam, you know. And then we had a final meeting with a smaller group at a meeting in Poland. And I had just been hiking in the Dolomites for fun in Italy. And I was in a wine producing region. And I found a bottle of wine that was called Porphyra. And I thought, this is too much. So
Yeah.
this very tall guy who's a German, Martin Lohr, I told him that he needed to bring 6 bottles of Porphyra wine for our final celebration.
Of course.
He arrived with his back on, bent back and back because his backpack was so heavy. We had a lot of
But
fun.
was it a red wine?
It was a red wine. Yes.
Thank you.
Yeah. And really tasty.
That's great. So let's talk about some of the algae that get where they're not supposed to be, and what they, what do invasive species mean for some of the biodiversity of areas, and how damaging are they, and what can we do?
Non-indigenous species often push out native species, not always, but that's often an outcome, and not one that we usually appreciate. And I notice I left off a little line here, so it should read at the bottom. So I was talking about old invasions of North America, Fucoserratus in maritime Canada, and others are very recent, like Grottolupia turuturo, which is a red alga, and you can just forget the sun. So at any rate, if you go on to the next slides, we have some slides. So this is an herbarium sheet that we pressed from Grottolupia turuturo, as we found it had jumped from a previously reported invasion that had gone up as far as the New Hampshire shore, all the way to mid-coast Maine in the Damariscotta estuary. And there are many ways it could have gotten there. There was actually an importation as much as 20 years ago, I guess, of Japanese oysters to try to start an oyster aquaculture. And Grottolupia didn't appear then, but of course the climate has been changing. So the Grottolupia could have just been hunkering down in that spot and bloomed. And there's now a thriving oyster aquaculture industry within the Damariscotta estuary of native oysters. And so it's possible, that some of that happened, and some other types of Asian oysters have also been imported. So we don't really know how the Grottolupia turuturo came in, but we found it on a class field trip at the University of Maine's Marine Lab on the Damariscotta estuary. So, it's always good to teach. You never know what's going to turn up when you take students into the field. And this is Grottolupia growing on some lines near an oyster farm. And yes, and so one of the things that was interesting to us is that the shellfish industry uses bleach as a disinfectant to prevent invasive species. You know, people are trying to be careful. And so we decided to see if it would work against Grottolupia and some native species. So here's your dulse, Liz. This is Palmaria. These are all in seawater, and this is after our bleach treatment. Well, you can see the bleach treatment pretty much takes care of dulse. It doesn't completely kill all of Grottolupia, and our native Cystocarps in the intertidal zone was just fine. If you go to the next slide. But the remarkable thing is that here's the Grottolupia, and there were all these little pustules that didn't get killed by the bleach. And it seems as if the carpospores, these are all what we phycologists call cystocarps.
Right.
So it is where fertilizations had occurred. This is a female blade, and spermatium, which is what we call red algal sperm, had fertilized the egg cell, and then it starts to divide up. Algae invented cloning before we did. And so the fertilized egg divides up into lots of spores. But here the spores seem to have fused, and out of each one in the recovery in seawater, a branch grew out, or it sprouted. And so this is what this looks like, you see, as all of these are starting to form. And then you get something that looks like this. And then these new blades get to look very large. And we found tetraspores on them. So this bleach treatment actually seemed to help spread, or had the potential to spread Grottolupia. And I guess I should say potential, right? This is an experiment after all.
Right.
Okay, this is the older invasion of Fucoserratus. And in studying this, Lad Johnson, who's at Laval University in Canada, helped, and Jim Coyer, Janine Olson, Gilles Haro, and Vitsa Stam, who were all working in the Netherlands at that point. Janine and Vitsa headed a very productive lab at Groningen, and April Blakesley. And so we were studying, if you'll go on, the invasion of both the periwinkle snail, Littorina, as well as Fucoserratus. And they were known to have probably come from someplace in Europe, but most people thought that that the snail had either been brought over by the Vikings or maybe the French from Brittany.
Been going on for a long time.
That's right. But I love history. And so I realized that the time period in which Fucoserratus and Littorina were found in North America coincided with a huge immigration from Scotland. And it was because the English, had kept the Scots from being able to really practice their culture. They couldn't wear kilts and so forth. And there were many more serious problems. So a lot of Scots came to Canada and the US in the late 1700s and 1800s. And the first ship to sail to Pictou, Nova Scotia from Scotland was the Hector in 1773. And so thinking that maybe both Littorina, the periwinkle snail, and Fucoserratus, the rockweed, could have been co-introduced, I started doing historical research. And I think some of that's shown as we continue. Here's Fucoserratus. And so this is exactly what you were talking about, Liz. There's just one Fucoserratus here, and this is the native Ascophyllum and native, this is probably Fucus disticus here, which is also native. And now in places like this, everything is Fucoserratus. So
wow.
it has really pushed out other species in the low zone The native species are still above. Okay, so I started going through old newspapers from the early 1800s and looking at where ships had come from to Pictou. And that led us to places that we should go sample Fucus and periwinkles in order to have DNA from sites where ships came from,
Right.
and see if there would be a match. And I'm pretty sure that this area in Loch Broom in Scotland is where the Hector sailed from. I don't know it for certain, but I'm pretty sure of it.
There it
Makes
is again.
sense.
Yeah.
And the open sea is out at the end here. And so this is all Fucoserratus.
Wow.
There are quite a few introductions with the new shipping that is taking place since the 1950s, 60s, 70s. I mean, presently they say 90% of everything goes by ship of products and goods. And along with the intended cargo, there's often the unintended cargo. And especially when you're wrap oysters in seaweed, which was a common practice, and it still is to some extent. So how did some of the 200 or so non-native species get to San Francisco Bay? Well, we talk about them being the invasives, but we are the source. We're not only invasive ourselves, but we've taken over the world basically in places where no humans were native until the 20th century and now into the 21st, and displacing the creatures that once did not have us to contend with.
Right. But it is really interesting how, through the historical research like this, we can go back and really kind of get a grip on
like a
where
detective story.
it is. It's
great.
To try to figure out, like,
where did you come from?
Where did they come from and how long have they been here?
Yeah.
Right. Yeah, so this is the reading room in the National Archives in Edinburgh. And both I got to examine customs records and shipping logs from the 1700s and 1800s.
What is the date on this? Can you see it? 1807?
Something like
Up
that.
at the top?
Yeah.
Right,
The 5th of July.
1807, the 5th of July. And
Yeah.
so this is the brigantine Active bound for Pictou in Nova Scotia.
Huh.
This is why it's important. This is why it's important for kids to still learn cursive, so they can go back and read these things. You can actually go back and look at the historical record and understand it.
Mm-hmm.
Yeah, it's true.
Fascinating.
So I put together over 800 records of ships by going through the Nova Scotian and Scottish records and some newspaper records from Ireland. Okay, Sylvia, so you have to talk about vivarium at Harvard.
Yes, got my fingerprints all over it for years. Contributed quite a few samples that were prepared like this. Press the juice out of anyway, dry them, as you say, desiccate them so they can be stored. And it's amazing that add a little water, you can sometimes not only, at least with some of the so-called blue-green algae, but are actually blue-green bacteria, they can come back to flourish. I mean, they never did quite die. They just went into a dormant stage. I don't think that's possible with the big kelps, but who knows? There may be spores that persist. And anyway, it's a record.
Yes.
Like pages in a book. The living pressed seaweeds are really a source of information.
And they are. I remember we had Barrett Brooks, who's looking at some of your old specimens at Smithsonian, and he was so grateful that you had just press them in the traditional way instead of trying to fix them with formaldehyde or do anything like that, because he was able to go back and take very discrete samples and pull DNA from them.
Right.
So it really did help to understand, like, tracing back some of the genome work. So it's
That's right.
it's a tried and true technique.
A little smelly.
It's not that bad. I mean, we had it in the garage, we had it in the kitchen, we pressed algae everywhere, trust me. I was recruited early on.
Yeah.
I know on my trips around the world, I always sort of feel sorry when I see a housekeeper in the hall, not knowing what I'm about to do to the room.
You have to leave a very large tip at the end, right?
Yeah, exactly.
Well, we're kind of creeping up to the top of the hour, and I think we should maybe go on to some of the questions.
Oh, no, there's more.
yeah.
Wait, what?
There's still more.
You think we have more slides? I know we have more slides, but
Well, maybe we can just zip through some and Susan can say, this is really something I want to touch on.
okay.
All right, right?
So
All right.
we can go slowly through this. Well, this will be archived, so people can go back and look at the slides we don't have time to talk about, and they can always write me if they want to, right?
Yeah.
Absolutely.
Yes,
So-
let's just keep going through so people have time to ask questions.
Well, this does talk about that ballast water.
Okay, so this is
Yeah.
not ballast water. So absolutely, since, in the 1900s, especially ballast water discharge is where larvae and spores introduced a lot of non-native species. But that wasn't the ballast that was used to keep ships right earlier than that. Instead, rock was. And so, people would have taken rocks for ballast from beaches just like this. And you see this Fucoserratus on a rock, and there's snails, periwinkle snails all over. And they could easily have stayed alive during the crossing. And then the rock ballast was thrown onto the shore. And I took this photo of a little Fucoserratus growing on the back of a periwinkle on Thanksgiving Day when I was in Pictou. And it's funny because obviously Canadians don't celebrate Thanksgiving, but I really felt as if my Thanksgiving had come when walking down on the shore. I picked up a periwinkle and a fucus tied together in that way. And so
Wow.
there are many tombstones in the Pictou cemetery of the original immigrants from Scotland, and we're looking at one.
Oh, love these stories.
Exactly.
And this is just an assignment from our DNA work on Fucoserratus. And we found there had actually been 2 different introductions into Nova Scotia. The one at Pictou came from Galway, Ireland, or someplace close, although Limerick is also close to that source population. And Greenock, Scotland, which was one of my more harrowing collecting sites, was actually worthwhile because that's where the Fucoserratus population came from that wraps around Cape Breton Island. Now, the amazing thing is that Harvey, who was a very famous Irish phycologist, do you want to say anything about him, Sylvia?
Well, just that it's a cornerstone. I mean, he accomplished so much that a reference point because he was so prolific.
Right.
And go ahead.
So Harvey named the red alga Pikea after Captain Pike, who on his voyages would collect algae and send them back to him. And so in Newburyport, Massachusetts, Captain Pike picked up Fucoserratus. And it's not on the American shore. Probably a ship had come in from Pictou, and there was a little bit in an anchor, but it didn't stick. But the interesting thing is that Captain Pike was part of a group of New York really conservationists and botanists who introduced the house sparrow to North America.
Deliberately.
And they had to do it twice. The first time it didn't take. So they brought more house sparrows over from England the next year. So this is just to say that attitudes about non-Indigenous species have changed in the last 170 years.
You certainly have. There's stories about trying to get the New England lobster established in California. And they take live lobsters by train and turn them loose, hoping that they would prosper and reproduce.
Well, we have the striped bass,
Striped bass is one of the few exceptions. Most of them didn't make it,
Yeah.
but striped bass took off. I think there may be more in Pacific waters than there are in its point of origin in the Atlantic.
Yeah.
So
It's funny
Yeah.
because, it used to be a little tougher to get an invasive or a non-Indigenous species to move about, on a rock or something like that. But now it can just be something as innocuous as a plane trip, and you can inadvertently, bring insects
Yeah.
or plant materials
well,
somehow,
there are many more people traveling
Yeah.
and taking, plants. Let's think about look across North America, how many natives have been displaced by ornamentals that have been deliberately, and you find them in nurseries.
Oh, I know,
They're
right?
from all over the world. So we are truly agents of change. Once upon a time, and even now, some of it is incidental, but we deliberately mix things up. Even coconut palms, they're carried by people everywhere, and other vegetables.
And the ocean.
Yeah,
Yeah.
right. Anyway, so we're troublemakers.
We are troublemakers, but not nearly as much trouble as bacteria, right? Those guys really mix it up.
Well, these bacteria are super important. It turns out that not only can we not do well without our bacteria, our microbiome, but the algae can't. And so we're beginning to understand that there's certain particular species of bacteria that an alga has to have in order to develop normally. And so this is a really nice experiment that Weiss did in Thomas Wichard's lab in Germany. And there is a culture of this alga, which is an ulva, in azenic culture. That means that antibiotics and things have been used to remove all the bacteria. And then they inoculated those little, just formless masses of cells with Roseovarius, and they started to get some normal structure in C with Meribacter, and they got some normal structure, but neither one was really the normal ulva. So when you put the two together, then you got these very nice little holdfasts as well as the normal development going on. So we're beginning to figure it out.
It's so important to, all those that are interconnections that we can finally be able to, get a grip and understand today.
Well, back to Harold Humm, he was best known for his work with algae, with seaweeds, but he started out, as an undergraduate, he majored in English, and then he moved to bacteria. He was a bacteriologist for his master's degree, and I think also for his PhD, but he was very versatile. He was more of a broad-based scientist than most are today.
And above all, he remained curious.
Yeah, exactly. And inspired it in others.
Yeah.
So here you're looking at.
So I often think of Humm because I have started to study marine bacteria too. In fact, very seriously for about 5 years. And so in this case, we extracted the DNA from tips of 3 different species of Fucus that are in different levels of the intertidal zone on the Maine coast. And then we analyzed their microbiomes. And this little diagram over on this side is a ordination diagram. It separates how similar or different individual samples of bacteria are. For example, this is from the low zone Fucus disticus, and this would be one sample, in this case of holdfast tissue, but we also did reproductive receptacles and just vegetative tips. And what you see is that every alga has its own microbiome because they are separating into different groups. And we thought that this was likely as much an effect of the vertical stress gradient that the bacteria on the algae are experiencing, just as different algae grow in different levels of a shore. And so if you go to the next one, Liz. Right.
All right.
So I know time, we really want people to be able to answer questions. So I'm just going to tell you what this means.
Okay.
So this is again another ordination, but here we did an experiment. So we did a transplant in which we moved some of the mid-zone fucus back into the mid-zone as a procedural control. And we made little clumps as we transplanted them. They're tethered into little anchor rings. And then we moved, this is a clump that is up in the high zone, and we watered it every 30 minutes during low tide for 3 weeks, which was quite an operation because we had many sites across the shore. And
I can imagine everybody's running about, watering.
So this is published by Charlotte Quigley and Kyle Capistrant-Fossa, who were two of my students working on this project. And here's Fucus spiralis, which again is the high zone, is most stress tolerant. And you can see that the dry Fucus vesiculosis transplants cluster with them. And here is the Fucus vesiculosis in the mid-zone, the normal one. Here are the procedural controls, which should be the same, and you see they are. And when we watered the clumps, their microbiomes didn't turn into something that was similar to the microbiomes of Fucus spiralis. Instead, they stayed like the microbiomes of Fucus vesiculosis. And then again, Fucus disticus is different. And these are environmental samples, bacteria from the seawater and rocks. So we could alter the microbiome by changing the amount of stress. So it showed that it's not just the different species that makes the Fucus spiralis microbiome different from Fucus vesiculosis, different from Fucus disticus, but it also is the bacteria directly responding to this amazing stress gradient over just a few meters in the intertidal zone. So just as biologists for well over a century have gone to the shore to understand how any organism develops mechanisms to resist stress, we now can do it for the bacteria. They're reacting in the same way.
Yeah,
Cool.
that's very cool, because I mean, stress is one of these things that, we're all subjected to it. So many species are subjected to it. And as climate shifts and changes,
Yeah,
the
it's
stressors increase in really interesting ways. So it's
a new world.
definitely is.
And bacteria, the microbes, the little guys, are looming large, much larger than we ever knew. I mean, imagine not even having a microscope to know the existence of bacteria. I mean, we are so fortunate to be where we are in time, to be able to see what others in our past just could not.
All right, you guys, we're going to the questions. We're going to keep Susan a little bit past 4.
Oh, it's fine.
Okay, good. Okay, Caroline says, my question is, does Susan know Jim Carlton? He did a lot of work with algae when I was in graduate school.
James Carlton.
James Carlton.
Yes, I know Jim Carlton, and he also has done a huge amount of work with marine invertebrates. In fact, he redid Light's manual to the invertebrates of the Pacific coast. And he is really Mr. Invasive Species. The most recent thing he did was to study both the invertebrates and the algae. There was a group of people involved, of course, that came to the West Coast after the tsunami during
Right.
the Fukushima earthquake and so forth. And so all this debris came loose with the tsunami and it came over to North America. So he had a nice cover story or two in Science on that work.
That, the impacts of that tsunami have just been so incredible. In one of the deep water ROV projects that we did recently, we came across a trail of teddy bears on the seabed that were just kind of plunked down there randomly, and they were like 4,000 meters of water. It's just the strangest thing. You're like, well, it's super creepy.
The invasive bears.
Invasive bears, but they kind of wonder, is somebody going to use this as a substrate for something? What's going to happen to them over time?
Bacteria will move in,
consume
for sure.
them, but yeah, you do see some, and those are those kind of really weird, sudden, just planetary things that happen that can just create big shifts in where species get to, even without our help.
There are other questions here.
Yes, Caroline is asking, when cyanobacteria were captured, was the photosynthesis ability a symbiotic one at first, and then it became an organelle?
Ooh.
Yes, I think we can be sure of that. And there are several more recent evolutionary events that are like what we call primary endosymbiosis, namely the capture and integration of a cyanobacterium into a chloroplast by cells, maybe 2 billion years ago. So the one that's more recent is about 90 million years ago, and it's called Pollinella. And it is a testate amoeba that has captured a cyanobacterium. And we can see different levels of reduction of the free living cyanobacterial genome. So Pollinella still retains more genes in what is on its way to becoming a chloroplast, but it's not completely there yet.
Again, we're so fortunate to have the capacity to dive in and see phenomena of this sort. The technology, well, you could say technology cuts both ways, that when it's caused a lot of problems for the natural world, but generally speaking, the ability to understand and see and know what was unknowable before we had the capacity to really do what is now being done to the chemistry of life, and see these associations with the depth of time, and see, who are we? Where do we come from? Who are all these other creatures who share space with us? And how does it all come together? How much of the natural world can we lose because of ignorance, not knowing? And yet being able to ask questions that we couldn't ask, let alone find answers until right about now. Yeah. Hoo-ha. Lucky us.
Prasad is asking, how much cleaning and processing is required to use algae that's intended for eating?
Oh, because of all the stuff we put in the ocean?
Well, I think he's thinking they're more about, like you were talking about, some of the little epiphytic critters that get on there, and
It's just
and how is it cleaned before it's packaged for consumption?
Well, of course, one avoids areas that have conditions in which there would be a lot of things attaching to algae that you're going to harvest. And then any wild harvesting company, and for that matter, the aquaculture companies too, have to have a sorting and washing apparatus, right? And so things are very well taken care of. They're just not, picked out so that you would go crunch and get a barnacle while you were eating an algae.
A little sand, a little periwinkle. Yeah.
In Asia, it's almost like they have huge washing machines with fresh water. And so the Nori crop is put in there and washed, and then it is lifted out and is put on a drying rack, which is going to make the Nori sheets we eat. They go under a little toaster.
Oh, that's great.
That's when the red turns to green.
Yeah, yeah, because it's changing. It's kind of almost kind of cooking it slightly, I guess.
Yeah, it's just like, it retains the chlorophyll because that's inside the lipids of a cell. But the really beautiful red and red algae is from pigments called phycobilins. And they are very water soluble and quickly broken apart by heat and things like that. So that
Good
they wash
reason
out.
not
They wash out, yeah.
if you're going to use the ocean to grow algae to eat, it's a good reason not to contaminate them with the kinds of things that we allow to flow in the sea: pesticides, herbicides, plastics, microplastics, nanoplastics, all those things that now haunt us. It's a good reason to shape up.
So we have a question about codium as an invasive species. What was that impact on native of the codium? What was the impact on native species?
So the nickname for codium in the Long Island area is oyster thief.
Oyster.
Yeah.
Codium is one of these amazing algae that is actually one cell, but it's huge. And so there are no real cell walls in between, and there's cell streaming of, nuclei and chloroplasts to and fro. But it doesn't look like it's one cell. It looks like it's lots of cells when you look at it because it's so big. At any rate, it attaches to any hard substrate and then starts to grow. And so it becomes very buoyant, and it could literally lift the oysters off the bottom and just float them away, which is why it's called oyster thief. And there's a lot of codium that has come into other areas of New England at this point, including in southern to even mid-coast Maine. And especially after a storm, just huge amounts of codium can be thrown up on the beach. It's out of sight most of the time, but it's really covering a lot of the bottom, and it is removing a number of native species, at least in terms of your normal biomass.
It seems like it could kind of outcompete them, and then maybe it shades things out too, because it's like if it's
Big,
so
bushy,
big,
yeah. But not all codiums are big and bushy. In fact, of the many dozens of species around the world, I guess only in warm and temperate. I don't know whether they're in any of the Arctic or any of the polar regions, but another of our friends, longtime heroes, Paul Silva, that was his specialty. And during times when I would go off on expeditions, come back with little shreds of codium, I'd give them to Paul and I'll ask him, "So, can you tell me where I've been based on the codium?" Without telling him whether it came from Australia or
cruel.
But the cool thing is he could do it, because they are specific. Well, some of them are more general in their distribution, especially with the introductions facilitated by us. But others are homebodies. They're quite restricted.
Even just like small disc, right?
Yeah, little lumps.
Yeah,
little branches.
Anyway. For anybody who lives in the New England area who's interested in invasive species replacements, a young professor named Jennifer Dykstra at the University of New Hampshire has published some beautiful papers showing the change in the native flora in that region because of waves of different non-indigenous species. So they could look up those papers.
We're going to grab two more questions.
Okay.
All right. Martin is asking, what role do the algae or seaweeds have for blue carbon sequestration?
Lots.
So half of a primary productivity in the ocean is, or I should say, half of primary productivity on the planet is due to algae. And so a lot of that is phytoplankton, but more and more of the marine forest of brown algae are being found to play a very important part in primary productivity, the carbon fixation too. And the trick is to get the carbon really sequestered in the deep sea vis-a-vis climate change. Because if it's taken up, but not taken out of circulation, it's not a permanent carbon sequestration. Of course, all these algae are producing oxygen. The blue-green bacteria are fixing nitrogen, which we need. We can't, all this nitrogen gas is around us, but to get the nitrogen we need for proteins we get from bacteria. So I think that the most obvious way in which things are taken out of circulation in shallow water might be the coralline red algae. What do you think, Sylvia?
Yeah. And
Because then you have
course,
calcification.
right.
And the haptophytes in the ocean that are also calcified.
There are two things that the photosynthesizers feed the non-photosynthesizers. I mean, all the little guys in plankton rely on the phytoplankton for their groceries, and then they get eaten by the somewhat larger creatures and on up the food chain. So the carbon value of whales that derive their nutrition, like all animals, from some photosynthesizer along the way, but long and twisted food chain, $1 trillion in terms of the carbon value of whales. And when the whales die, they go to the, that's sequestration. They go to the bottom of the ocean.
Right.
But it's true with tuna. It's true with squid. It's true with all life in the ocean, all the animals. Ultimately, historically, before we started taking them out by the ton, that carbon went to the bottom of the ocean. And there's the blue carbon comment that Liz refers to. It's life in the ocean. And if you keep the carbon there instead of hauling it out by the ton, that's a part of what has held the planet steady for lo these many gazillions of years. And it's one thing that we could do, leave more of the fish, the squid, the shrimp, the lobsters, and other creatures in the sea, and that which we take out, well, yum, yum, seaweeds. Yeah.
Indeed. And so I want to mention, I think it's in the chat, that especially people who are looking for fun things to do with algae at parties and also in classes, if you go to this link to the PSA's site that Gigi has put in the chat, www.psaalgae.org. If you go to the education tab, you can find all kinds of things to do that are fun at parties, but and in classes. But this is a bookmark, and here you see a beautiful diatom. So diatoms are especially a large component of the total algal photosynthesis, and they grow in freshwater systems. They even grow in deserts. So, these are bookmarks, and they're all explained if you go to the PSA site. So algae make up desert crust, and some of those are diatoms as well as the cyanobacteria. It keeps them from eroding. But diatoms have silica valves around them, little glass valves. And so when they die, they rain down towards the bottom of the sea. And coccolithophthorids, which are parts of the haptophyte algae, they are calcifying and they will rain down to the bottom of the sea. And so there are many ways in which the algae are involved in sequestration. And the points you make about how we've interfered with the ocean ecosystem are very important too.
You're here.
Okay, this is going to be the last question. Ready?
Okay.
Are you both ready?
Roll of drums.
Ready the drum roll. What is the one thing you want people to know about marine algae? What's the one big takeaway about marine algae?
Other than they taste good.
Other than they're tasty. We know they're tasty.
Yeah. Well, we really do rely upon them for our well-being because, as I said, half of the photosynthesis that is involved in oxygenating Earth and also drawing down carbon is being done by algae. So they're extremely important organisms, plus the large brown kelps provide the structure that many other organisms use as habitat. So they're just tremendously important in the coastal zone as a habitat.
If you like to breathe, you'll listen up.
In fact,
they
in my classes, before I really start talking about photosynthesis and where it evolved in the algae and so forth, I have a class of students. I have a stopwatch in my hand and I say, okay, I don't want anybody to faint, but take a deep breath and hold your breath and see how long you don't have to breathe. Raise your hand when you have to take a breath. And hardly anybody gets beyond a minute. And I say, okay, you better be really grateful to the algae today. That's where your oxygen's coming from.
That's right. Give them some due credit, right?
Yeah.
Absolutely.
What fun to have a chance to talk with you, Susan. We need to go out and splash around together again.
Indeed. This is
And
really
I brought a piece of algae swag with me that
Ooh.
I have to see if I can get one for you.
We
Oh,
will.
One of our friends made
Great.
a Gatham tote, So algae is being appreciated, I think, in more ways today than ever before, which is a really outstanding thing. So
Yeah.
thank you
And-
so much.
Yeah. Thank you.
Thank you.
It's so much fun to talk with you. And thanks to
Yeah,
everybody for listening in.
And before we close off today, we just want to say thank you to all of our guests and our producer at the Ocean Elders and everyone in the community who keeps coming back and diving in with us. It feels like home to us, and I hope it does to you as well. Water connects us all, and we're so grateful to everyone. And until next time, remember, take care of the ocean and the algae.
And the algae, as if your life depends on it, because
It does.
it does.
It does. Thank you, Susan. We'll see everyone next time.
Yes. Bye-bye.